I'm Professor Dame Pam Shaw. I trained in neurology at the University of Newcastle, and when I was training, I just felt that I saw lots of patients with different neurological disorders, but motor neuron disease, I used to almost dread seeing a patient with MND in the neurology clinics, because really it made me feel like a useless doctor. We knew nothing about it, what caused it. We did very little in those days to be able to help patients facing that cruel disease or their families. I sort of felt it was a neglected condition and that we should try and do something about it. So that's how I got interested, really, from the point of view of just feeling it was a neglected condition. So I'm not sure there really is a typical working day for me. I'm a clinician scientist, so I see patients, and these days my neurological practice is largely devoted to MND. So I see patients in the clinic. I get queries every day about patients from the clinical secretaries over in the hospital, but then I have a big research team, and there are multiple research groups working on MND within SITraN, and we collaborate together. So there's a lot of busyness from the research side as well. So it's balancing those things, and emails. Of course, I get about 150 emails every day, so every spare minute is spent trying to keep on top of that email influx. So it's busy. Days are different. Some days we're doing scientific research, sometimes clinical research, clinical trials and so on, sometimes actually looking after clinical patients in the hospital. So it's very busy, very varied, but I can honestly say I'm never bored. And, I think doing both sides of the coin, the scientific work and the clinical work, means that one drives the other, really. Seeing the patients drives your enthusiasm for the scientific research, and I think scientific research benefits from the willingness and help that the patients give us, the patients with MND and their families, that they give us in terms of donating biosamples, being willing to take part in clinical trials. So it's a wonderful life, really, but very busy juggling both those sides. So I think my favorite parts are when you or your team make a significant discovery that kind of moves the state of knowledge along in terms of really trying to do something about motor neuron disease. So an important discovery that may make a difference to patients is really one of my favorite things. But I think the other favorite thing is helping early career researchers fulfill their potential. So, helping them through their PhD or through their postdoctoral research projects, getting nice publications, nice presentations at meetings for those early career researchers. So I think findings that may eventually improve the outlook for MND patients, and bringing along the next generation, helping the early career people, blossom in their working life as well, are my two favorite things, I think. And my least favorite things are when again, there's probably a couple of them, when bureaucracy kind of really slows us down in terms of the research that we're trying to do. I think we are very caught up with a lot of bureaucracy that slows us down. And obviously, one of the things that you have to learn as a researcher is to try and be resilient. And we all hate it when our paper gets rejected or our grant application gets rejected. And especially when we feel that maybe one or two of the reviewers have either been unfair or haven't understood the application. So you have to dust yourself off when those things happen. And I think you sort of have to learn to recover from those setbacks and keep going. I think have been the unraveling of the disease mechanisms in MND. So initially emerging from genetics, so identifying different genes that can cause MND and then modeling those genetic variants in cellular models or in vivo models to enable you to really dig down into what it is that's damaging the motor neurons. There's been a lot of progress from that side of things in the last few years. I also think that some of the therapeutic approaches that have come through, where our teams in SITraN did some of the fundamental work enabling those therapies to come to fruition for patients. So two examples are survival motor neuron, SMN replacement for spinal muscular atrophy. That's now a licensed treatment for those young children, infants really, that have a form of motor neuron disease caused by SMN deficiency. And then the real trailblazer is the tofersen treatment for the subgroup of MND patients who have a change in the SOD1 gene. So I've done, I think, 25 clinical trials for patients with MND, and the tofersen series of trials is the first one where patients have been reporting after a few months of treatment usually that sometimes stabilization of their condition, but sometimes actually improvement in muscle strength. I'm getting stronger. I'm doing things today that I couldn't do 6 months ago. And the other really positive thing from the tofersen antisense oligonucleotide trials is that it enabled us to see that measuring a biomarker in either blood or cerebrospinal fluid, neurofilament proteins, which are released when motor neurons die, and the tofersen trial showed us that if you've got something that's working, the neurofilament levels decrease in blood and CSF. So at last we might have an early indicator that a treatment is working, and I'm hoping that may make our future trials more nimble, quicker to do, quicker to decide if a treatment's working and if it's not, moving on to a separate approach. So very exciting times, I think, therapeutically. I think the area that is particularly exciting at the moment is the prospects for developing better neuroprotective treatments for MND patients. So that translational neuroscience side of things, which we work very hard on in SITraN. So, the trailblazer, the treatment for SOD1 related MND, shows us that we can, because we know there are more than 30 genes that can cause MND. So SOD1 has been the trailblazer, that we may be able to apply a similar approach for other genetic causes of MND. But for MND more broadly, for those 90% of patients who don't have an identifiable gene change, then that lower the biomarkers. So really the genetic therapy trials have shown us you need a biomarker to show target engagement, to show that the treatment you're trying is hitting the target properly, but also a biomarker of treatment efficacy. So if you've got a treatment that's working, hopefully we should be able to see that it lowers the level of neurofilament proteins. And we've got a platform trial set up called Experts ALS that's testing 3 different drugs at once, aiming to find those ones that lower the neurofilament levels. So I think it's a whole new ballpark in terms of therapy development. And I think the other very positive and hopeful thing is how interested pharmaceutical companies and biotech companies are in finding treatments for MND patients. And I think that's because of the trailblazers. They see now that if you direct the right treatment to the right patient group, you can treat motor neuron degenerative disorders. So I think pharma companies see MND as an attractive model of neurodegenerative disorders. So, a few years ago, pharma companies weren't very interested in MND because it wasn't a big enough market size, or it wasn't perceived as a big enough market size, but that's rapidly changed. And I really believe in productive collaborations between academic institutes such as SITraN and pharma companies, and they're coming to us all the time wanting to test their promising molecules in the model systems that we've set up in SITraN. So I'm very aware that the pace of medical science does not seem fast enough if you're a patient or a family facing that distressing condition, but I think things are moving very fast, and I personally am optimistic we'll have better therapies available for patients in the quite near future.